organism is. An organism is a living thing. And so moving forward, we're going to talk about, we've started talking about kingdom Protista and kingdom fungi, and now we're moving on to talk actually about the interaction between living things. And so we're going to start with the interaction between microorganisms, micro meaning small, so small living things, and the complex multicellular organisms that have immune systems. So we will focus a little more in the next section on what the immune system is. We're starting first in 9.1 by talking just about what are examples of microorganisms, starting by talking about prokaryotes. And so prokaryotes are really the smallest and the most prevalent organisms. So we seem to discount them because they are unicellular. But that doesn't mean they're not an important part of biology. A prokaryotic cell, we know by breaking up what the word means, that pro means before and kary means nucleus. So these are cells that we believe to actually be what could be called relics or the ancestors of other cells. And this is where abiogenesis came into play. When we studied evolution and we looked at what could have been the origin of life, we looked at simple cells. And the idea that the simplest cell might just have four things. That the simplest cell is going to have a cell membrane. That it is going to have cytoplasm. That's the inside of the cell, and the cell membrane separates us inside and outside. We also are going to find DNA and ribosomes. And remember, the importance of DNA and ribosomes is that from this, we can build proteins, and proteins can perform certain functions. So when we look at what a prokaryote is sometimes called a simple cell.
Caleb Hickman, if you're still on campus, please come to the front office. Caleb Hickman, please come to the front office.
But what we want to focus on is that doesn't mean that it doesn't meet its own needs. We can't dismiss prokaryotes as unsophisticated, because what we're going to see is they do actually have fairly complex functions. This is an interesting idea of the prokaryote in terms of being an example of unity. That this could possibly be where life arose from, and that we're going to have a lot in common with prokaryotes. We've actually already gone over a lot of examples of unity of life. When we start talking about prokaryotes, we do want to also focus on what makes them different from other living things. Remember, prokaryotes and eukaryotes, these are really the idea of the domain. But keep in mind that prokaryote does include the domain eubacteria and the domain archaeabacteria. So when we talk about prokaryotes versus eukaryotes, we're not trying to give more prestige to one or the other. We just want to honor the fact that they have different adaptations. So prokaryotes are going to be unicellular. They can live in a colony. They can communicate, but they don't require multicellularity, meaning that a single cell is still considered an organism. For eukaryotes, we do have some that are unicellular. These are the protists and some fungi. But many are multicellular. Some fungi, definitely plants and animals. When we look at cell size, often we'll say that prokaryotes are very small. Eukaryotes are known to be 10 times larger. So they are going to be categorized as a smaller cell. However, there's an advantage to being a smaller cell. When you have a smaller volume, you have a larger surface area ratio, which means you can perform functions a lot faster. So it's not that there's necessarily an advantage to being larger and a disadvantage to being smaller. Half dozen of one, six of the other. When we look at the chromosomes, keep in mind chromosomes are coiled or packaged DNA. Prokaryotes are going to have a single circular chromosome, but they will have extra chromosomal DNA, known as plasmids, which we'll talk about a little more later. Eukaryotes are going to have linear chromosomes, so this should be a little bit of a review. Prokaryotes are mostly going to perform binary fission, but we are going to see something today called conjugation. You might have heard of conjugal visits back when they used to allow inmates to have their significant other come for a visit. So conjugal actually refers to sexual reproduction. We're going to see that bacteria actually have a way to swap genes. When we look at eukaryotes, we are going to primarily see, so primary is sexual reproduction. But of course, we do have an importance of asexual reproduction, and that's for growth and repair. Next, we talk about internal compartmentalization. Prokaryotes really have no internal compartmentalization. Eukaryotes do. They have a lot of organelles that are membrane bound. Meaning that the membrane is actually going to create a specific compartment. We do see a trend of something called a flagella, which is going to be important for movement. And prokaryotes have a very simple one protein flagella. Eukaryotes are going to have a much more complex flagella. So when we look at things like the euglena in the protist or the sperm in the animal, we're going to see a much more complex arrangement of proteins. For metabolic diversity, we are going to see a lot in the prokaryotes. We will see a lot of metabolic diversity in the eukaryotes, but it tends to be broken into kingdoms. So we actually tend to classify organisms based on their metabolic diversity with our eukaryotes. So as we move on, we're next going to look at the two different types of prokaryotes. So we'll look at the archaea versus the eubacteria. So you do need to know how prokaryotes and eukaryotes are similar and different. We also want to know how eubacteria and archaea bacteria are similar and different. So one of the things that we'll notice is that both of these are prokaryotes. And so for a long time, they kind of belonged together. And then they started to research and they realized that prokaryotes are actually two very different lineages. And so we've got two lineages that are believed to have split millions of years ago, if not billions of years ago. And archaea is actually known to be more similar to eukaryotes than archaea is similar to eubacteria. We do just want to make a note that even though they both have cell membranes, archaea have different lipids. So we're going to find a different lipid composition. Eubacteria have what's called peptidoglycan. Archaea do not. Eubacteria, when they translate their genes, we're just going to have different ribosomes in the archaea. And we are going to find introns in archaea bacteria. So this is the reasoning for why archaea bacteria is its own domain. Next, we need to move on to talk about, are bacteria really that simple? We call them simple cells, but it turns out that there's a lot more to them than meets the eye. One of the things is the cell wall, and you might want to pause here, but the idea is that in eubacteria, we're going to have two different types of cell walls. We're going to have what's called a gram-positive cell wall and a gram-negative cell wall. Gram-positive is going to absorb and show a certain stain called a gram stain. And this is because the gram stain attaches to the peptidoglycan. So what we start to do is we start to group prokaryotic organisms based on their anatomy. And of course this can lead to some problems with phylogeny. But one of the ways that we'll group them is based on how their cell wall is constructed. Is the peptidoglycan on the exterior, or is the peptidoglycan actually covered by an outer membrane, which means the stain can't attach to it. So that's one of the unique characteristics about eubacteria. They start to break into two groups. Next, we want to look at the cell interior. The cell wall is going to be the exterior. The interior, what we're really focusing on is the idea of actually the DNA. The DNA is the most important part. We are going to see that the DNA is the singular circular chromosome, but we will have what's called a nucleoid region. However, it does not have a nuclear membrane. We'll also see the idea of a plasmid, which will become very important in just a moment when we talk about trading plasmids. The ribosome, we do also just want to make note, is very different from the eukaryotic ribosome. And this is actually one of the targets for antibiotics. The thing that is kind of most unusual and unexpected about prokaryotes is actually that we do find variation. We've talked about variation in terms of sexual reproduction, but bacteria don't reproduce sexually. They reproduce by copying exactly what's there, and that's asexual reproduction. So where does variation come from? One of the things that we'll see are mutations, but the bigger source is actually plasmids. And it turns out that they're actually able to exchange plasmids through a structure called a pili, and this is known as conjugation. Conjugation. J-U-G-A-T-I-O-N, conjugation. And so what we see here is the ability to copy your plasmid and actually share it with another bacteria. And that's very unexpected here. Not something that we really had known until we started to study them on the cellular level. So we'll talk about the implications of this in terms of sharing successful genes. Real quickly, we do want to talk about the idea of metabolism, and we said that this is very diverse. We can be autotrophs or we can be heterotrophs. We do want to keep in mind photoautotrophs are actually believed to have produced the first oxygen when we talked about the evolution of the first photosynthetic cell. And then we have chemoautotrophs, which are the organisms that are able to use chemosynthesis, and they do not need light. So these are the ones that are the sulfur-consuming bacteria at the depth of the ocean. They will cause disease, which we'll look at, one of which is dental cavities, or known as caries. We'll talk more about that. And we do also want to know that some bacteria are very important in that they can be beneficial to other organisms.